Hi, Chris Good here. This is another Video Essentials lecture, and the topic today is neuroanatomy. So, this is the schematic of your brain. It's a cartoon image, and you can see that this is the scalp that lies over the skull, and beneath that is this space, including some vascular tissue as well. And then underneath that, you see the brain proper. This is the outermost part of the brain. It's the bumpy part. It's the part that most people recognize as being brain. We call this the cortex, and underneath the cortex are some other brain structures that we're going to be talking about today, including the brain stem that extends down till just inside of the skull, the cerebellum behind that, and there's some other structures I'll point out as we go along. We're going to start at the bottom here in the brain stem, work our way up to the cerebellum, the midbrain, some structures in the central part beneath the cortex, and then we'll talk about some cortical regions, and then I want to mention just a couple of neurotransmitter systems and show you where they're located in the brain. Okay, the brain stem has two major parts, the medulla oblongata, this is the longer part down here, and the pons, which is this bulgy part up here. Now, the brain stem's the source of a lot of vital autonomic or automatic body functions. So your breathing rate and your heart rate is governed by neurons whose cell bodies lie here in the pons and medulla. Also, any sensory information that's coming in from the outside has to go through the brain stem to get up to the rest of the brain where you experience that sensory input. All of these little nervelets, these little roots that you see poking out here, contact structures in the head. We call them cranial nerves because they innervate parts of the head. They send motor commands out to muscles that move parts of your head around, including your mouth, lips, and tongue. And they receive sensory input from all of your head holes, except for your nose. So your eyes, your ears, your mouth, all of the sensory structures around there go into the brain stem via these cranial nerves. Here in the pons, you see the autonomic respiratory centers. This is the part of the brain that tells other neurons that innervate the diaphragm what the breathing rate should be. And this happens automatically without you having to think about it. But there are certain drugs that have depressant effects like alcohol, barbiturates, opioids that can suppress activity here in the brain stem and drive respiration down to the point where you can go into respiratory arrest. You'll stop breathing and die. So this is a very important part of the brain. Just remember that everything that's coming up the spinal cord from the rest of your body, going down the spinal cord out to the muscles that move your body around, has got to go through the brain stem too. So this is just another view of the brain stem with it turned toward you. Just on top of the pons, right, which is on top of the medulla in this picture, you see the middle part of the brain, or the midbrain, and just on top of that you see these two egg-shaped structures to the left and right at the very top of the image. This is the thalamus. The thalamus, it does a lot of different things. It's often described as a relay center for sensory information that's going to the cortex. It does so much more than that. It's involved in modulating motor output with the rest of the basal ganglia. It's involved in modulating sensory input that's coming into the cortex, and it plays a role in some limbic functions as well, and we'll talk about that in a minute. If you turn the structure around, look at it from the side again, you see the brainstem again, the pons, the medulla, and a little cerebellum on the back. Cerebellum literally translates as little tiny brain, and we know that it's involved in modifying motor actions, including the adaptation of certain reflexes to changing environmental conditions. The cerebellum gets sensory input from all over the body, and it integrates that sensory input to fine-tune motor actions so that they get the desired result. It's important for learning ballistic movements of your body, like jumping or throwing stuff, so that it improves your accuracy with iterations of certain motor actions. When you get damage to this area, it can result in a severe lack of coordination that we call ataxia. And so there are certain drugs that act on the cerebellum that, if they interfere with its function, can induce that lack of coordination. Alcohol is one of them. Just above the brain stem and the cerebellum lies the middle part of the brain called the midbrain. I want to call your attention to it in this aspect because it's you've got a really good view. Right where it's pointing, this arrow says mesencephalon. There's a part of the next brain region that we'll talk about called the basal ganglia, called the substantia nigra, and it lies just about right where that line is pointing. I just want to call your attention to the fact that the midbrain is split into two parts by the mesencephalic aqueduct that is a fluid-filled space that sends fluid down here into what's labeled as the fourth ventricle. And so that splits the midbrain into a part called the roof, or the tectum, that here is labeled corpora quadrigemina, and the tegmentum, which is the floor. It's everything on the other side of the aqueduct. And right where it's pointing there, mesencephalon, right in this dark region back there, that's a part of the midbrain called the substantia nigra. And it's seen here in this aspect as part of the basal ganglia. You don't need to know all of these parts, like the putamen and the caudate globus pallidus, and parts of the thalamus, which all contribute to modifying motor output so that our body movements are accurate and precise, and that our body is moving where we want it to go. The substantia nigra plays a part in this as well. It's called substantia nigra because when you look at it with the unaided eye, you can see that these cells that again are involved in motor output are very, very darkly stained. And these are the cells that are selectively targeted in Parkinson's disease. These are the cells that disintegrate, they deteriorate as a result of Parkinson's disease. In fact, on the right, this is a normal brain stem, midbrain. And on the left, this is from a Parkinson's patient. You can see that just with the unaided eye, that those darkly stained cells are almost all gone here in the midbrain of the Parkinson's patient. Interestingly, these are dopamine-containing neurons, and we're going to come back to that in a little bit. So just remember the spot, substantia nigra. There's another brain region very close by that also involves dopamine cells in the midbrain, and we'll talk about that when we discuss transmitter systems in just a moment. Just a couple more systems I want to talk about. The schematic over here shows you where the limbic system is, right? So there's part of the cortex is considered to be limbic. This is the cingulate gyrus in yellow. And then underneath that you can see some other brain regions, including parts of the thalamus. This is the hippocampus that curves around up above the thalamus and then out into the temporal lobes and is deep inside the cortex of the temporal lobes. And at the very end of the hippocampus, there's the amygdala. And let's see, the pineal gland located behind the thalamus and behind the hippocampus is also considered limbic. As is a region that's not pictured here, is just underneath the thalamus, called the hypothalamus, and this I believe is the pituitary extending down in front of that label. So the limbic system we know to be involved in emotion and motivation, learning and memory, some basic and yet very complex types of behavior require the limbic system to be functioning normally. And we'll talk about how some certain drugs can act on limbic regions to change some of these behaviors. Finally, we're going to talk about this outside, the outermost part of the brain. This is the cortex. Cortex is an anatomical term that's used all over the body, not just in the brain. Cortex literally means bark, and like the bark covers a tree, the cortex covers the outside part of our brain. We usually divide the brain into 4 different lobes. The temporal lobe, which is, if you imagine this as a boxing glove, then this is the thumb, right, of the glove that's sticking out there. The very frontmost part is called the frontal lobe, and it's separated from the back by this, it's a line that's labeled central sulcus that is kind of wavy and it extends all the way down to the temporal lobe where it says lateral fissure. Pretty much everything behind that up to the very backmost part of the brain is the parietal lobe, and then the very back of the brain is the occipital lobe. And really what you should understand about this is, about the cortex, is that this is where we experience the sensory world for a lot of different senses. When you feel pressure on your body, it's because of activity here in this post-central gyrus, the primary somatosensory area. When you see something, it's because of activity here in the occipital lobe. And if we stimulate these regions of the brain, you'll experience sensation in a different part of your body, a different sensory modality, depending on where we stimulate. One last thing, I guess, these two halves of the cerebral cortex are connected to each other by a very thick body of fibers that is called the big body, which is what corpus callosum means, and you see it sort of severed here in this aspect. That actually connects the left and right hemisphere of the cortex so they communicate with each other. A great deal of fuss has been made over what behaviors occur in the right hemisphere and what behaviors occur in the left hemisphere. The fact of the matter is that we have two hemispheres. We have one whole brain, and it's connected, and this is one of several points at which the left and right brain are connected, and they work together to produce behaviors. I think that the distinction between left brain and right brain things is often overblown. If we look on the underside of the brain, we get a really good view of the blood supply to the brain. Your brain needs blood. That blood contains oxygen. The oxygen gives the cells the energy they need to keep running those sodium-potassium pumps, to keep nutrients flowing, to keep transmitters being catabolized, to keep the brain working. And it has an incredible blood supply and demands a great deal of the body's energy. We'll talk in a different lecture about the blood-brain barrier, that where only the stuff that's in the blood that your brain needs is able to get out. All right, we're going to wrap this up by looking at where a couple of different neurotransmitter systems are. I think it's very important to realize as we talk about these different neurons that are releasing different neurotransmitters that there's no single transmitter system that governs individual behaviors, that all of these transmitter systems interact with each other to produce complex behaviors. So just keep that in mind as we go along. So the first thing we'll talk about is neurons that make acetylcholine. Those neurons' cell bodies lie here in the basal part of the forebrain, and their axons project all over the cortex. So even though the acetylcholine's being made here, those neurons are releasing acetylcholine here in the hippocampus, up here in the frontal part of the brain, down in the occipital lobe, the parietal lobe, all over the place. And there's other neurons here in the midbrain where there are acetylcholine-containing neurons whose axons project into the cerebellum, into the pons, down into the medulla. Acetylcholine goes everywhere. And it's these neurons, acetylcholine neurons, that are often suffer widespread loss in Alzheimer's disease, and new drugs to treat symptoms of Alzheimer's are targeting these cholinergic pathways. And again, acetylcholine is the transmitter that's produced by these neurons. Next are two dopaminergic pathways. We've already discussed one of them, and the substantia nigra. So the cell bodies that are manufacturing dopamine, transporting it down their axons and releasing it in axon terminals, those terminals are located in parts of the basal ganglia, and some up here in the pons as well. We call this the mesostriatal pathway. So it goes from the midbrain, and it's also called the nigrostriatal pathway. It goes from the substantia nigra part of the midbrain up to the globus pallidus and other parts of the basal ganglia that have a striated or striped appearance in certain brain sections. So it's the midbrain to the striatum pathway, substantia nigra to striatum. And remember that this pathway is important for motor function, that it's what gets destroyed as Parkinson's disease advances. And right next to substantia nigra is the very bellymost or ventralmost part of the tegmentum, which is the floor part of the midbrain. The roof up here is the tectum. And this ventral tegmental area has neurons that release dopamine at the ends of their axons here in something called the nucleus accumbens, here in the hippocampus, and up, and so these are limbic structures, right, and part of the limbic system, and then on up into the cortex as well. So we call this the meso-limbo-cortical pathway. It starts in the meso, the midbrain, goes to the limbic system and to the cortex. This is probably the most relevant brain pathway for discussing drug use and drug abuse. This is also referred to as a reward pathway, I think erroneously. It's better to call it a reinforcement pathway. When you allow laboratory animals to self-stimulate this pathway by inserting electrodes into it, and you can read about the history of those experiments online. I'll link to it via the blog. That's all they'll do. They'll just sit there and do whatever the task is that gives stimulation to this pathway. They will self-stimulate, with a physiologically relevant electrical current, right, that makes these neurons fire anywhere along this pathway. They'll do that to the exclusion of any other behaviors. They won't eat, they won't sleep, they won't drink, they won't have sex. They'll just stimulate this pathway. So it's not very surprising to find that all drugs of abuse ultimately stimulate this pathway, whether they affect dopamine neurons directly or not. Cocaine, amphetamine, methamphetamine, these are drugs that work directly on dopaminergic systems, but alcohol, nicotine, opioids, marijuana, these activate this pathway indirectly. And yet people compulsively use the drugs, right, that send this signal to do it again. Now, why would we have this pathway that is so susceptible to drug addiction and stimulants by these substances? Well, it's not there so that people can become addicted to drugs. It's there to teach you how to do things that are good for you. So this pathway is also activated when you eat a good meal, when you have sex, when you take a risk and there's a chance of winning something. This pathway is activated. So it teaches you to do things that are good for you in the sense that it's promoting your genes, right? It's passing the genes along to the next generation. We've found ways to hijack that pathway and stimulate it without having to do those behaviors. And there's some behaviors like gambling and compulsive, sex addiction that stimulate the pathway too, and it sort of gets out of hand for people. But the pathway is there to teach you to do things that are good for you. All right. Just a couple of more things that we want to talk about. Serotonin is a transmitter that many people understand is involved in mood because drugs that are used to treat mood disorders, like antidepressant drugs, target serotonin. Not all of those drugs target serotonin specifically, only the most recent ones do. And there are serotonin neurons all over the brain. Their cell bodies lie in nuclei spread throughout the brain stem and up into the midbrain, and their axons release serotonin onto cells all over the cortex, into the limbic system, into the basal ganglia, into the hippocampus, the cerebellum, down the spinal cord, the pons, and so serotonin neurons project everywhere, and they're receiving projections from other cells. So to say that serotonin is a transmitter that makes you happy really reveals a misunderstanding of how complicated mood is and how much these systems interact with each other. We'll also see that when we talk about antidepressant drugs, that's just not the case. You can't give serotonin to someone and have their mood change instantly. It's actually a more subtle effect than that. And we'll come back to that later on. I don't have a picture of where glutamate is an amino acid, and it's the principal excitatory neurotransmitter in the central nervous system. And that's why I don't have a picture of where they project. There are glutamate neurons everywhere in the central nervous system. And there are many different types of glutamate receptors. I don't want you to try to memorize all these different kinds, and we're not going to talk about their function just now. Just understand that glutamate is typically an excitatory transmitter, that it has many different receptor types that end up basically stimulating the postsynaptic cell in an excitatory sort of way, and we'll come back to it later on. Finally, gamma-aminobutyric acid, or GABA, is the major inhibitory neuron in the brain. There's lots of different types of receptors of GABA. We've characterized just a couple of them. Instead of opening a channel as the acetylcholine receptor does to pass sodium, when GABA receptors open after binding their transmitter, GABA, it's a negatively charged ion, an anion, the chloride anion that comes into the cell, even hyperpolarizing that postsynaptic cell even more, driving it further away from where it's going to fire an action potential. And drugs that interact with GABA receptors increase this inhibitory effect of GABA. So these are drugs like the barbiturates, benzodiazepines like Valium and Xanax, Klonopin, and alcohol as well works this way, as do all of the current second generation sleeping pills. They activate GABA receptors so that they're increasing their inhibitory influence on postsynaptic cells. All right, we will see you in the next lecture.